This video is Unit 6 review for the EOC. This is on genetics. And genetics, again, very important unit of the year. We spent quite a bit of time on it, so we're going to spend some time going over it as well. Just as a quick review and foundations for our work in genetics, there were a couple of names that we mentioned that you should be familiar with. One of those was a Frederick Griffith. And I'm just going to basically lay out what he's known for. Remember, he's the one that did the experiment with the mice and learned that bacteria can actually transfer DNA between one another, and this causes bacterial transformation. Now, Griffith didn't know that DNA was the change agent, but he had his suspicions. And so it wasn't until later that DNA was found to be the carrier of hereditary information and the change agent in this. But he just found out that live bacteria that were harmless could be changed by dead bacteria that were at one time harmful. Griffith's predecessor was a man by the name of Oswald Avery, and Avery repeated Griffith's experiment, and he isolated DNA as the change agent. Now, what's difficult about this is that in this time, many believe that proteins were actually the change agent in these sorts of RN cells. And so Avery was kind of going against the grain in the world of science. And so two people by the name of Hershey and Chase, Alfred Hershey, Martha Chase, definitively concluded that DNA is the molecule that transmits hereditary information. And here's the way they did that. They used bacteria. And with these bacteria, they also used something called a bacteriophage. And a bacteriophage is a virus that kind of has a funky alien landing craft look to it. And what these viruses do is they basically they land on the bacteria. And let me zoom in a little bit here. They will actually land on the bacteria and inject their DNA. So if this is the, let's say this is the viral or the bacterial DNA, well, the virus will actually inject its own DNA into that bacteria, and that DNA has the code to make more viruses on it. Well, eventually the bacteria will replicate and make more and more pieces of the virus until that virus just kind of, or the bacteria just kind of explodes, and all those viruses go out and infect other cells, and that's how a virus spreads. Well, they thought they could use a virus because viruses are made of two things: proteins, which are the outer shell that you see there, and DNA, which is on the inside, because these are the two molecules that are in dispute. And so here's how they did that. They used two elements that are found in one element found in lots in proteins, and one that's found a lot of in DNA. So for the protein test, they used sulfur, and they marked it radioactively so that it showed up almost like I don't know, like glow in the dark almost. If that's the way you want to look at it. Not really glow in the dark, but you get the idea. All right. And it had the DNA inside of it again. And when these bacteriophages infected the bacteria, they injected their bacteria or their DNA into the bacterial cell, and through subsequent generations, what they found is that none of the none of the radioactive sulfur was found in the subsequent generations. They just kept getting bacteriophages or bacteria that look like this, normal bacteria. And so the radioactive protein stayed with that original bacteriophage. Now, in the next case, they used radioactive phosphorus. Phosphorus is an important ingredient in DNA. And so we have this kind of glow-in-the-dark DNA. Now, what they found here is that in subsequent generations, the glow-in-the-dark DNA was found in that same bacteria over and over again. And so this shows that the DNA, not the proteins, is what is being passed from one generation to the next. And so Hershey and Chase definitively concluded that DNA is this transfer molecule. Very important in the history of genetics. So, what is this molecule DNA that we've been talking about? DNA is made up of nucleotides, which we have talked about in Unit 2. A nitrogenous base, 5-carbon sugar, deoxyribose in this case, and a phosphate. And DNA is made up, as far as these nitrogenous bases, there are A's, adenines, T's, thymines, C's, cytosines, and G's, guanines. And a man by the name of Chargaff found that the A's and the T's were always in equal percentages, as were the C's and the G's. So much so that if you knew the percentage of one, you could solve the percentage of all the rest of them. How do you do that? Well, if you know that there's 20% of the cell DNA contains adenine, well, you also know that 20% contains thymine, or 20% of the nitrogenous bases are thymine. Well, that leaves 60% left. Divide that by 2, you've got 30 and 30. So that's one way that you can use Chargaff's rule. And so this went a long way to helping to figure out the structure of DNA. And a lady by the name of Rosalind Franklin took a picture, and this picture was done by a method called X-ray crystallography, and it essentially showed it's basically, if you can imagine taking a DNA strip and looking at it from the top down, and it showed this X pattern. And this X pattern, Franklin suggested, meant that DNA had a helical shape. Now, Franklin died before she was able to finish her work, and so two men picked up her work by the name of Watson and Crick. And these guys were the first ones to make three-dimensional models of the DNA molecule and showed definitively that DNA is indeed helical in structure, and it is not just helical, but it is a double helix. So you have one molecule and another one that kind of wraps around it like that. So we've talked about DNA chromosomes, or we've talked about chromosomes, prokaryotes having a single chromosome, eukaryotes having many chromosomes. The prokaryotic chromosome is typically circular, whereas eukaryotic chromosomes are linear in nature. And before we begin talking about the essential dogma concerning genetics, we need to talk about the difference between DNA and RNA, which is single-stranded, which is one of the big differences there. So the three main differences between DNA and RNA is single-stranded, double-stranded. RNA is single-stranded. DNA is double-stranded. DNA has deoxyribose and RNA has ribose as the sugar. And RNA uses uracil, whereas DNA uses thymine. So in RNA, anywhere that there would be a thymine, there's instead a uracil. And there are three kinds of RNA that we're going to look at. First is a messenger RNA. The messenger RNA is a temporary copy of a gene. All RNA originates from DNA, but messenger RNA has a particular role in protein synthesis, whereas it just lasts long enough to be translated into a protein and is then degraded. rRNA makes up ribosomes, or is part of the makeup of ribosomes along with some special proteins. And so ribosomal RNA. And then there is pRNA, which is the transfer RNA. And transfer RNA transfers amino acids to the ribosome during protein synthesis. There are other kinds of RNA, but we're not going to get into them. Now, real quick, something about DNA replication that we need to mention. DNA replication is quite complicated, but for our purposes, we're going to keep it very simple. All right, not Watson and Crick, but DNA. All right, so this is DNA replication. DNA replication involves the DNA having to be split in two. And so the first thing that's going to happen is the DNA is going to kind of be relaxed. And so the way this works is there's an enzyme called helicase, and that helicase will actually separate the DNA structures to where they're kind of like that. And you'll have your P's and C's and G's and A's and T's and T's. And so on the other side, you have the opposite structure. All right. And so helicase is this big protein that is just unzipping the DNA. Well, and that's, let me write helicase down. Helicase. Another enzyme called polymerase is actually coming back behind and placing new pieces of DNA. Separate these here a little bit more. New nucleotides next to the DNA. So like this. Where there's an A, or where there's a T, it'll put an A. Where there's a C, it'll put a G, and so forth. And it's doing that on the other side as well. Where there's a T or an A, it'll put a T, and you get the idea. There's a mutation. We'll fix that real quick. All right, and it does that for all 6 billion of your base pairs until the entire thing is replicated. And so if you start with something that looks like this, what ends up happening is each strand is being copied and a complementary strand is being made such that by the time you're done, you have two identical pieces of DNA with one old strand and one new strand. This model is called the semi-conservative model of DNA replication. Now, to move on to a more complicated piece, we're going to talk a little bit about protein synthesis. All right. and we can move through this pretty quickly. Protein synthesis is the production of proteins. And my goal here is not to give you a really in-depth overview, but more so to give you kind of what the basic overview. Protein synthesis has two stages: transcription and translation. Transcription, the goal of transcription, this happens in the nucleus, and the goal of transcription is to make a messenger RNA. Make a messenger RNA using or from the DNA. So all proteins, protein synthesis is the making of proteins. All proteins originate, the information for these proteins originates on the DNA. That is important. All right, and so that messenger RNA will leave the nucleus and go to a ribosome, and there on the ribosome, the messenger RNA is going to be used to make a polypeptide or a protein from the messenger RNA. And that's translation. Translation. And individual amino acids are brought in order to be joined together on the ribosome until you have this long chain of amino acids called a polypeptide. And like we talked about in Unit 2, that then long chain will fold up and make a protein. Proteins have shapes, and those shapes do particular jobs. And so this is how you get from the DNA, where all your information is, to making an actual protein, which does the work. Now, again, much more complicated than that, but hopefully keeping it at the upper level here, and you guys can go back and review any details you need to. And same thing here as we talk about quickly about meiosis. I want to just briefly go over this. I want to make another video with Mendelian genetics. But meiosis is the production of gametes. And this is sperm cells in males and egg cells in females. And a couple things I want to get with meiosis is that there are two divisions, but only one interphase. One interphase where the DNA replicates, and then you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then you go immediately into prophase 2, metaphase 2, anaphase 2, telophase 2. And so let me just briefly go over this. So if this is prophase, we're going to just use, we'll start with interphase here. In an interphase, we have a chromosome and its homologous pair there. All right, and this is a very simple cell. Well, in prophase 1, what's going to happen is those chromosomes are going to replicate. And those homologous pairs are actually going to get close together to where you have this structure that is called a tetrad. And when these tetrads form, it can make it to where the actual chromosomes will swap pieces. And so you can end up with, using my same colors, you can end up with a piece that looks like this, and a red one that has a little green piece on it, and then one that is normal. Oh, that's enough actually. All right, and so that's called crossing over, and that happens during prophase 1. All right. Now, during metaphase, or so during prophase one, they line up there in the middle. During metaphase one, or during prophase one, they get together and form tetrads. During metaphase one, they actually line up in the middle. So I won't draw a separate slide for metaphase one, but you get the idea. Pretty simple phase. But anaphase one is where something simple or serious happens or important. And this is where the homologous pairs are separated. And so the cell is no longer a diploid cell at this point. And so using my same chromosomes from up here that I've doctored a little bit, they separate. They pull towards the ends. The centrioles pull them just like in mitosis until you end up with telophase 1, cytokinesis. You end up with two haploid cells like so. Now, next, there is no, again, there is no metaphase, or there is no interphase 2. That's important because the DNA does not replicate again. The DNA just simply stays like it is. And so during prophase 2, nothing specific happens. Metaphase 2, they line in the middle. And so during anaphase 2, these sister chromatids will separate telophase 2 and cytokinesis so that you have 4 cells at the end, each one of them haploid, and each one of them genetically different from the other. This one here should actually be red. All right, and so these are haploid cells, genetically different from one another, and these end up becoming the gametes.